Can you say something?
Yeah, can you hear me?
Yeah.
Perfect.
Okay, let's just wait a second. I need to put you on the big screen.
Okay.
Okay, our next speaker is Feargal Ryan. By the way, welcome after lunch. And he's calling from South Australian Health and Medical Research Institute from EMBL from Australia. And he is going to be talking about application of machine learning techniques for creating urban microbial fingerprints. Okay, Feargal, please go.
Thank you very much, and good morning, everyone from South Australia. So it's just 4:30 a.m. here, so forgive me if I'm a small bit slow this morning. I'd really just like to start by thanking the organisers. I really wanted to attend the meeting, but unfortunately it wasn't meant to be. But the organisers have really just been so helpful and friendly, and really great in getting everything set up. So thanks a lot to them. But anyway, let's dive right into it. So, like I said, I'm Feargal, and I'm speaking from South Australia Health and Medical Research Institute, SAMRI, but I actually just moved here. So when I began working on this, I was back in the south of Ireland, County Cork, where I was based in the APC Microbiome Institute. And then since May, I've been over in Australia. So I think you've heard a bit about this over the past couple of days, but the data I'm going to be presenting you today is based on the MetaSub CAMDA challenge. So the MetaSub consortium, as I'm sure you all know, but if there's anyone that just joined or came in after the break, this is the metagenomics and meta-design of subways and urban biomes. So according to the website as of yesterday, there's almost about 4,000 samples taken worldwide, which is very impressive. I first became aware of this cohort, or well, this type of project anyway, back with PathoMap when it was published in Cell Systems in 2015. Again, very impressive, very impressive study. But obviously now with MetaSub, this has really been built out into this huge worldwide network of metagenomics samples examining the urban microbiome. And I really think that this is an absolutely just fantastic resource, not just for examining the urban biome, but as I show in a bit, also for mining for novel genomes and novel genomic sequences. So moving on from there, the CAMDA challenge, again, as many of you have heard, there was a few hundred samples from several different cities in different countries around the world, from North America, South America, Africa, Europe, New Zealand, and also in Japan as well. These samples were also collected from a variety of different surfaces: wood, metal, plastic, rubber, although primarily from metal, which proved a bit problematic, which again I'll talk about in a bit. And also in New York and Sacramento, they had both samples included from the pilot study. I just think it was published as part of PathoMap back in 2015, and also from a newer study, which I'll talk about also in a bit, because this proves to be a bit tricky, especially for New York. So I'm going to talk, or well, here I'll just give a brief overview of the analysis framework that I ended up going with for this. So there's kind of two branching forks here. The first you'll see basically quality control of reads, taxonomic classification. The tool I chose to do this with for this was Kaiju. And I'll talk about why I chose Kaiju in a minute. But basically what I did with this was I just built a large database from the inner database, or you took the inner database and basically essentially almost every genome there. The human genome, all plant genomes, fungi, viruses, archaea, bacteria. And then from this built a single database, classified all reads against that, and then essentially used this to generate a count table where you have each feature representing a certain taxonomic rank and the number of reads assigned per sample at that feature. And then this is what was used for dimensional reduction, classification, random forest, and the results I'll show you there in a couple of minutes. I'll come back to the assembly bit in a while, and well, based on my timing anyway, I don't have a huge amount of time to talk about that, but I'll do what I can in a while. So why I chose Kaiju for this? 3 reasons. First, speed. It's very fast, considering that it takes each of the reads and does a 6-frame translation, which is very, very impressive. Because it does a 6-frame translation, it's really looking at amino acid homology, which makes it sensitive to more distant relationships through this translated searches. And the third reason was stability. Generally, when I'm working with this type of metagenomic data, that is metagenomic data where the majority of sequences, or at least most of, or a large portion of sequences anyway, are not going to be represented within reference databases. I generally don't work with reference databases. I just like to do de novo assembly and work with the metagenomic OTUs or MOTUs, as they've been referred to in some studies. But here, that was maybe a bit tricky because I knew that there was going to be the initial set of samples, and then because of the CAMDA challenges, the mystery samples, they were going to be added in later on. So this rose the kind of prospect of stability, which is something from the 16S field, and this was more in the concept or in the kind of the realm of de novo generated OTUs, that clustering sequences in 97%. If you add in or take away samples, the OTUs themselves can change because certain sequences are now added or they're taken away. That can also kind of be the case with metagenomics, because if you're assembling more samples, you're adding that in, suddenly you might have new metagenomic bins, or a certain metagenomic bin might get bigger or smaller, and that might change what you're thinking of previous samples. So I just wanted to try and avoid that problem for this. But ultimately, as I say later on, I really do think this type of data would really benefit from a de novo assembly, a really comprehensive de novo assembly approach. So this is what Kaiju brought back to me, basically at a very high taxonomic rank, kind of close to the main level. Animals, plants, fungi, bacteria, archaea, viral, and unclassified. And what we can really see here just from the get-go is that even at this high rank per city, you can still see really large differences. In all cases, the majority of classifiable sequence is bacterial. In some cases, actually followed by animal, although most of what's there is actually classified as animal was just human DNA. There was also small amounts of tardigrade DNA, which I personally thought was kind of cool, but we're not here to talk about what I thought was cool. Then we also have, then the classifications that were used for further downstream analysis were basically just microbes, fungi, bacteria, archaea, and viruses. I also included unicellular eukaryotes in this as well. So as I mentioned earlier, there was differences that I detected between the pilot samples and those that were generated later on. So if we can just highlight the two New York samples here, you can see this is the samples generated in 2016, and these are the pilot samples. And even just from this, and I won't go down into much more detail from that, you can see there's a fairly considerable difference actually. So when I really got down into doing my analysis, I decided to just work with the New York samples taken in 2016 to avoid any kind of batch confounders for the classification. So moving on from that, taking this table of features that are generated from Kaiju, obviously representing, I think the original table was well into the tens of thousands of features, which dropped down a bit after I filtered for anything above a certain abundance. I used TISNE, T-stochastic neighborhood embedding, to reduce dimensions down to 2 and represent it on that. And when I do reduce this down to 2 dimensions, we get this absolutely lovely plot. We really form these kind of discrete groups. And this isn't just a feature on TISNE. This was actually a few different methods that I tried produced quite similar results, which was quite nice and quite reassuring. So what we can see here is that most cities tend to form discrete units, except Auckland and Hamilton, 2 cities in New Zealand which don't, and they overlap quite a lot. C1, C2, and C3 are the mystery cities that were provided as part of the challenge, or for the second part of the challenge, I should say. I'll come back to those in a bit. Yeah, so what we can really see here is that the samples cluster nice and discretely by city, and that Kaiju features do seem to be able to discriminate them based on this. So I guess the obvious question from this is, microbially, what's driving this? And that's sometimes maybe a bit tricky to answer when you have such high dimensional data. So what I did was I just took what are the most abundant taxonomic ranks that were detected, and I just plotted out the top 12 of them. And even just at those 12 ranks, we can see fairly significant differences actually between the cities. Auckland, Hamilton, New York, and OFA have very considerable amounts of Pseudomonas. That's that kind of light lavender color, which is far lower than in Porto, Sacramento, Santiago, and Tokyo, which in turn have higher amounts of Plasmodium, which seems to be much lower in the other samples, and so on. So even at just this very high taxonomic rank, with just these handful of features, we can actually see these cities delineating a bit and separating out. We can also see that known proteobacteria, and I say known proteobacteria because I'm sure there's a lot of unknown proteobacteria that we're not capturing here, that these are far more abundant in the 4 cities there on the left-hand side of the plot, from New Zealand, New York, and then also OFA and Nigeria. So, on to next. So, building a random forest classifier from these features. Basically, what I expected it to return based on the plot in TISNE was that it was able to distinguish between these cities quite reliably. So this was built using 2/3 of the data and then tested on the final third. Excluding the New York pilot samples also had the added benefit of balancing out the groups a bit more, because before that New York was really the far largest group in the entire cohort, making very unbalanced for classification, which is something I'll come back to with the surface, or come back to when I'm trying to classify surface in a minute. So the main source of error here we can see is Auckland and Hamilton, which, as you can see from the TISNE plot and also in the just those stacked bar plots I show you, their profiles are generally quite similar, although not completely identical. Pretty much every other sample is classified as it should be. And yeah, so there's just a class error for Auckland and Hamilton. Auckland, the samples were evenly split between Auckland and Hamilton, and that was almost the same for both cities. So when I asked random forest what are the features that, when taken out of the random forest, they reduce the training accuracy the most, there was, well, several, but I'm just going to mention 2 here today in the interest of time. The first was gram-positive bacteria, Staphylococcus argentineus. So this was previously considered a lineage of S. aureus, and it was reclassified, I think, only 2 or 3 years ago. This is generally detected worldwide and associated with human skin and sometimes. And the relative abundance of this on the y-axis, we can see varies quite considerably by city. Detected at decent amounts in Porto and Santiago, a bit lower in Tokyo, virtually undetected in New Zealand and Africa by comparison. We look next, one of the other top features that was associated, or one of the other top features from the random forest for classification was this fungi, Conosporium apolinensis. So members of this genus have also been isolated from human skin. Sometimes it can cause disease, but generally it'd be asymptomatic. And again, we can see fairly considerable differences from this. In this case, this particular species was considerably higher in Sacramento generally than in other cities. And I guess really what we're seeing from this is that a lot of these very highly predictive taxa don't seem to necessarily be going on these surfaces so much as they're actually deposited possibly by humans moving through, which I think is probably quite key to what we're seeing here. And I think that's probably why, at least from my analysis, there wasn't great separation of groups by collection surface. And I think that would hold true if what we're seeing is really just the human commensals or bacteria from human touching being deposited onto it rather than live cultures growing there. So samples were collected from a number of surfaces, but this was, well, quite tricky to build a classifier from. Well, first, I think probably biologically, but also second, the samples or the collection groups are a bit unbalanced. Most were collected from metal. Some of the other specific groups, like vinyl, were only actually within one city. So then it makes it a bit difficult to dissociate the city signal from the surface signal. So moving right along onto the mystery challenges. So the first challenge, I'm sure you've heard plenty of on this in the past couple of days, so I'll breeze through it quite quickly. There was approximately 30 samples from different cities already featured in the primary data set, and the question was, can you identify these? Using this method and just clustering the samples closest to the ellipsis that they were plotted on in TISNE was generally yes. All samples were correctly identified, except the New York samples. I believe that this was because of the difference between the 2016 collection and the pilot, because the pilot samples clustered closer to New Zealand, whereas the 2016 samples formed a discrete cluster for further down closer to Sacramento and Santiago. And those are marked there with the arrows. On challenge 2, 3 cities not previously included. And these were labeled on the plot from earlier, C1, C2, and C3. C1, this is another city in Nigeria, and it clusters with OFA, which is quite nice to see. C3 is Boston, which is down there with New York and the others, and C2 is Lisbon, which clustered with Auckland and Hamilton, oddly enough. And then the third mystery challenge was 16 samples with no information about which city they came from. And again, this was quite nice to see that I was basically using this method able to predict generally which area that they were falling in the plot, and detect which samples that were coming out together, which was good to see. So just briefly to conclude on this section, the majority of detectable sequence is bacterial, which I think we already knew, although for most it's not at species level. And it generally seems for most that while we might be able to classify it to a certain taxonomic rank, we certainly don't have a genome for this in the database. But based on this alone, we can already separate out cities by homology to known phyla. Nearby metropolitan areas do seem to have overlapping microbial profiles. They don't seem to be identical. And I really think if we started to dig down into that unclassified, we'd be able to delineate these probably even more. And then surface type had less detectable impact than city of origin. Again, if I had to guess, I would probably put this down to the fact that what we're really seeing is not, as I said, live bacteria growing, but rather things being deposited from humans passing by and touching the surface. So very briefly, I'm going to touch on a small bit of work I did with some assembly approaches. So like I said, in the interest of time, I'm going to keep this short and only show a very brief overview. So what I did was I took megahit, assembled all the samples, and did a small bit of analysis to try and look into really what was there. So the largest content to come out of this, and these were assembled per sample, there was no cross-assembly done or pool assembly. The largest one to come out was 1.7 MB content. This showed basically almost no nucleotide homology to anything, at least in the NT database. As we can see by this figure there at the bottom, those little red dots showing conserved areas of homology and very little of the rest. So really what we can see of this is that there's quite a lot of these actual very large contigs on it that represent maybe 30, 40, 50, 60% of a bacterial genome, but a novel bacterial genome. Of the contigs over 100 kb, only 17% of them had a blast in hit to anything in the NT database, and that E-value cut off. And then down there we can see the linked distribution. But again, this was just assembling per sample. What I guess would have been a bit more comprehensive would have been assemble per sample, pool per city, and then perhaps take the reads from that don't assemble either, and then assemble those and then try a number of different binning techniques. I didn't do that here because I figured it wasn't really part of the challenge, but I think that there's a lot that can be done with this. So I'm just going to end with a very, very short story from the human gut virome field. My postdoc back in Ireland before I moved to Australia was in a gut virome lab. And this paper published back in 2014 was the first one to describe this cross-assembled phage, crass phage, which is a very highly abundant bacteriophage in the human gut. And what these people had found from Robert Edwards' lab was that this bacteriophage of 97 kb was in fact 2% of all human fecal metagenomic data that had been generated up to that point. Yet no one had ever described it, no one really knew anything about it. And so they discovered the sequence, that it was highly abundant, and they predicted it to have a host in Bacteroides. Skip on a few years later, what we can see is that Eugene Koonen's group describes this as actually one member of a very expansive bacteriophage family in the human gut. But again, up to this point, this is really all just based off metagenomic data and assemblies. And then recently, here in this preprint, people have actually now isolated this. And it does infect Bacteroides. And it basically has the life cycle that was exactly predicted several years ago from the metagenomic assemblies. And I think this is a real success story for metagenomics, and especially for that metagenomics of the uncultured, the unclassified portion. So I really think that it's these sorts of methods show that subdata is an absolutely prime opportunity to mine from novel taxa and novel genomes. And with that, I think I've taken enough of your time. Thank you very much for listening. Again, I really have to thank the organisers for their help and for being so kind for getting this set up. My contact details are down there, but I think if you just Google my name, there's not a lot of Feargal Ryans in microbiome research. So thank you all very much for listening, and yeah, thank you.
Thank you very much, Feargal. So I think we could try with one question. I'm not sure if this will be feasible, but does anyone have a very burning question that would like to ask? Okay, so I hope you can hear this.
I don't know if the mic's connected.
Let's try.
Hi, Feargal. Great talk. I'm curious on how many species, you showed some example plots where some species were very particular to very specific cities, and how many really good sort of city-specific marker species do you think you could find in data?
There was quite a lot. I don't know the number off the top of my head, but I mean there was, like at least in some of the tables I generated, there was somewhere between tens to hundreds. Actually, there was quite a lot.
It's a lot, yeah. That's great. It's good because a good fingerprint database then, yeah.
Exactly. Yeah.
Okay. Thank you very much.
Thank you very much.
And have a great day.
Thank you. Bye bye.
From States. Bye.
Bye.